Mutations. Woo. So this is our last part of this unit, last information. This unit again, when we look at it from like the beginning to the end of Bio 30, from Punnett squares to technically cell division, because that's technically included in part of this unit, even though I kind of like separate it, all the way to this mutation business, is about 40% of Bio 30. So it is a lot of information crammed into some small little bit of time. So we're gonna finish this. It's gonna be fun. I love mutations. I think they're super cool. And we're going to talk a little bit about how we can use our knowledge of mutations and our knowledge of DNA and kind of bring that together with understanding how organisms are actually related to each other based on evolutionary theory. I'm trying to move this so that you guys can see my board a little better. There we go. So mutations are just changes in the sequence of a DNA molecule. So again, your DNA has a specific code, and if there's any changes to that code, that can change the overall proteins that are going to be made from reading that specific code. So typically mutations are the source of any sort of genetic variation we have within a species, and these can then be acted on by natural selection. So if they're beneficial and they will help to promote that organism's fitness, that means that they will probably be passed on to future generations. When we're talking about fitness as well, please be aware that we're not talking about like how buff these organisms are, but how well they are reproducing. So the more fit you are, that just means the more reproductively successful you are. So 3 kind of general types of mutations that you've probably heard of before. We have beneficial mutations, which, surprise, benefit the organism. They're good. Harmful mutations, they're bad, okay? They're gonna harm that organism's chances of having a mate and doing its reproductive business. And the neutral mutations, you don't even know they're happening because they literally have no impact on that organism. So from these 3 kind of broad terms of mutations, we also have some more specific ones. So we have point mutations, which are happening at a very specific point in the DNA molecule. And then gene mutations, which are impacting an entire gene. So something that is coding for a specific protein, as we've talked about. So 3 types of point mutations. We have silent mutations, which guess what, are neutral because they're silent and we don't know they're happening. There are nonsense mutations, which don't make any sense, and missense mutations. So silent mutations, again, don't know they're happening. Nonsense mutations actually turn the original codon into a stop codon. And then a missense mutation is just turning one amino acid into a different amino acid. So silent mutations, again, they typically happen also in non-coding regions of DNA, which means that they happen in areas of DNA that aren't necessarily genes, which means that we're not transcribing them, we're not translating them, they're not actually coding for any sort of specific trait. Because again, all of your DNA isn't necessarily turning into something specific in your body, like your hair color or your eye color. Only specific segments in those gene pieces are actually being transcribed and then translated. So if these mutations are happening in a non-coding region, then they're not a huge deal because they're not turning into anything anyways. And that's as of right now. We think that DNA doesn't do anything, okay? Maybe in the future we'll find that that actually does have a big impact on stuff, but as of right now, it's not a huge deal. Or this mutation is actually changing a nucleotide to something else, but it doesn't change the overall amino acid that's being made, so it really doesn't have any impact. So in this picture here, our mutation that's happening is with this little guy here, so it's supposed to code for an adenine. Instead, we end up adding in a uracil, but we still create the threonine amino acid, which means that our protein chain doesn't change, so life is good, we don't know any different. Nonsense mutations are bad, because they stop the translation of a protein or stop the creation of a protein before it's fully made. So we're taking something that's supposed to be an amino acid and turning it into nothing. So these ones are typically lethal to the cell, which if you're a single-celled organism, that's really bad because, you know, if it's lethal to you, then you're dead. But if you're a multicellular organism, it's not the end of the world. Okay, so this guy here, again, same original mRNA strand up here, but down here we actually have 2 points that have been changed. So we have a U and an A being inserted here, which turns this part into a stop codon. Missense mutations, again, are taking a amino acid, swapping it out, and then instead of making the original amino acid you were supposed to make, you're making a different one, which could potentially lead to some issues. So a really good example of a missense mutation is actually sickle cell anemia. So your red blood cells are supposed to be nice and circular and biconcave and super awesome and happy, kind of like this little guy here, okay? This is a nice happy red blood cell. This is what they're supposed to look like. But a sickle cell ends up looking like these weird moon shapes or sickle shapes. Kind of like a sickle that they used in farming back in the day. With sickle cell anemia, again, a couple changes in the amino acid sequence change the overall structure of the hemoglobin molecules, which again are found in the red blood cells to help with oxygen and carbon dioxide transport. And by changing those protein shapes, it changes the overall structure of the red blood cell itself, which is going to impact the amount of oxygen and carbon dioxide that can be transported. And by having these seed kind of shapes, these sickle blood cells are able to kind of connect with each other and interact with each other and can end up forming clots and clumps and bad things in blood cells that we don't want to have happen. So in this case, again, our normal mRNA strand we have up here, and then we have a point mutation, which again, a single point on the DNA strand, where we're changing something. We're changing this guy right here, which means that we're going from a threonine amino acid to a proline amino acid, which again, may not change what we're making completely, but it could change the shape of it. And by changing the shapes of proteins, we're actually changing their functionality and how well they're actually going to work. Okay, so point mutations, there's 3 of them: silent, nonsense, missense. Gene mutations, we have 5. So these ones, again, are going to impact an entire gene. Point mutations can impact genes too, but we just call them different things because typically gene mutations are going to be looking at more than one nucleotide that's being changed. So we have deletions, insertions, frame shift, translocation, and inversion, which when we get into them, they're pretty self-explanatory as to what's happening. So with a deletion mutation, you're literally deleting nucleotides. So you're taking stuff away, which again is going to cause differences in the amino acids that are going to be made, which are going to cause differences in the overall protein. So remember as well that if it's one mutation, so because this is a deletion mutation, it may also lead to other mutations happening. So just because you have one mutation doesn't mean that's the only thing that's going on. A lot of times there's many, many other things happening as well. So in this deletion mutation here, we've taken out an amino acid, and that changes this threonine amino acid into a histidine, which again will change the overall protein. Insertion mutations, this is where we're adding nucleotides. So it's the same as deletion, but instead of taking them away, you're adding some in. This again is going to change the amino acids that are being made and change the overall protein structure. In this case, by inserting a U into this mRNA strand, we end up actually creating a stop codon. So this gene mutation, this insertion mutation, is also going to be a nonsense point mutation because we're turning an amino acid into a stop codon. So this is an example right here of being a gene mutation and a point mutation. Frame shift mutations are most mutations. So this is taking the reading frame, so the order of the amino acids, and changing them. So anytime we change an amino acid in a mutation, you're changing the frame shift, you're changing the reading order. So insertion and deletions are almost always going to be frame shift mutations as well. Translocation mutations, this is similar to like a transformation kind of experiment where you can take a piece of DNA and insert it into something else. It's the same kind of thing. A lot of times these happen where we have crossing over occurring, but between non-homologous chromosomes instead. So you'll have like chromosome 5 and chromosome 10 coming together and doing some crossing over business, which we don't want to have happen. I'm also in the process of doing some Google Meets with some of my other classes right now, so if I'm like pausing for a second, I apologize. So crossing over when it happens between homologous chromosomes, life is good because your genes are in the same position, you're just swapping genetic information to increase variability, but when we have it happening between non-homologous chromosomes, you could be trading eye color for chances of diabetes, for example, okay, which those genes aren't supposed to match with each other, so then we have some weird business going on, we have lots of weird proteins being made, and it's just awkward and not a good time. I'm just going to pause my video for 2 seconds. All right, so the gene mutations again, we've had insertions where you're inserting a nucleotide, or 2, or 3, or 4. Deletions, you're taking them out. Frame shift mutations is just changing the way that the amino acids are being read, so pretty much most mutations. Translocation, again, is where you're taking a piece of DNA from one area and you're like cutting it out, and then you're moving it over here and inserting it over here. So the amino acids that were supposed to be made over here are now being made over here, which again is going to disrupt that entire protein. Okay, there are some kinds of leukemia, so blood cancers, okay, that are caused because of these types of mutations. So mutations, a lot of times you're going to hear about when we do have any sort of conversations about cancers, because cancer, a lot of the time, is changing your DNA sequence that leads to uncontrolled growth of specific types of cells. And then our inversion mutation is taking a piece, cutting it out, and flipping it around. So this picture that I have for you here, the original good strand goes 6, 1, 3, 5, 2, 4, 7. Okay, 6, 1, 3, 5, 2, 4, 7. When we invert part of it, we're going to take out these 4 numbers here and flip them around. So now we go 6, 2, 5, 3, 1, 4, 7. Okay, so we've taken these numbers here, turned them around, and then put them down here. Again, this is going to change the reading frame, which is going to change the amino acids that are going to be made, which is going to change the overall protein, change the overall function, and again, could be beneficial, could be harmful, could have no impact. So the genetic material is still the same, it's still the same letters, it's just now backwards of what it's supposed to be. And some of the reasons we have mutations, they can just happen, okay? They're spontaneous mutations, they do happen. Sometimes replication doesn't go the way that it needs to. Sometimes our polymerase molecules aren't able to actually do their spell check job properly and they miss something, so they just happen by chance. Or we have induced mutations where we can actually cause a mutation to happen, which a lot of times are caused from outside environmental factors like UV radiation or X-rays exposure or other things like that. Okay, so spontaneous again happens because DNA replication just doesn't do its job properly sometimes. Okay, our enzymes aren't perfect, things can happen. Induced mutations again, because of mutagenic agents like UV radiation, like chemical exposure, like X-ray radiation, cosmic rays, any sort of those wavelengths on the electromagnetic radiation spectrum that are above or below our visible light spectrum can have impacts on our DNA. Okay, the web activity. So for this, I'm going to move you guys over here real quick. Am I going to get it the first time? Is it going to happen? Oh. Maybe if this remote would actually work. I did it. Woohoo. See, I'm kind of figuring stuff out slowly but surely. And then getting super fuzzy. Come on, camera. There we go. All right, so we're doing a web activity. Again, this is just some way to help you guys summarize the information and take a bunch of notes and kind of cram it into a smaller sort of section. So I want to make sure I'm writing this not in an awkward way. Mutations. Boom. Apologize for my wonderful sound effects. I'm also going to have to move my thing up a little bit so that you can see more of what I'm actually doing. Maybe. There we go. Whoa, way too far. I really apologize for the awkwardness of these wonderful videos that I'm sharing with you guys, because I'm obviously so good at all this technology business. All right, so with our mutations, we again have those kind of three general types. So you can be neutral. Can you see that? Yes. You can be harmful, or you can be beneficial. And these, again, are just very, very general types. Okay, so neutral, don't have any impact. Harmful are obviously going to harm that organism's chances of having more babies. Beneficial is going to increase the chances of that organism having some babies. Because again, everything in bio is apparently about having babies. We also have point mutations. And the three different types of point mutations, again, you can have silent mutations, you can have nonsense, or you can have missense. Okay, silent. This again is where you're taking an amino acid, or taking, a nucleic acid, not an amino acid, one of the base pairs, and you're turning it into potentially the same base pair. Or your amino acid just stays the same. Okay? And I will scroll that down when I'm done with this whole thing here. Nonsense mutation is an amino acid turns into a stop codon. And a missense mutation, again, is where you turn an amino acid into a different amino acid. So this is like taking a histidine and turning it into a stop, whereas this one would be taking a histidine and turning it into tryptophan. Write the right thing down. And then over here again, histidine stays as histidine. So silent, we don't know that it's happening. Nonsense, we're going from an amino acid to a stop codon. Missense, we're going from an amino acid to a different amino acid. I should be able to see that now. And then lastly, we have our gene mutations. I don't want to go too. And then gene mutations, the different types, again, we have insertion, we have deletion, we have translocation, insertion, deletion, translocation. My brain is not functioning. Excuse me 2 seconds because I apparently can't remember what I just talked about. Inversion. Okay. And all of these will lead to a frame shift mutation. So insertion, you are adding nucleotides. Deletion, you are deleting nucleotides. Translocation, you're moving nucleotides to a new location. And inversion, you are flipping your nucleotides backwards. And then again, frame shift mutations are taking anything that's happening to cause a change in how your amino acid change in your amino acid sequence. Or again, changing how that protein is supposed to be read. Because if we change the amino acids, we are changing the overall protein shape, which is changing the overall function of said protein. And so with our knowledge of mutations and how things are happening, we can then kind of take this out and look at the bigger picture as well, and we can actually see how mutations over time and changes over time can be linked through evolutionary history if we are looking at this from an evolutionary standpoint. So the last little bit here is looking at mutations in evolution. So biologists, again, are able to compare the genetic makeup of different organisms and different species and look at the relationships between them. So how similar are their DNA? Or how similar is their genetics? Phylogeny is the proposed evolutionary history of a species or group of organisms, and you can link the information you get from this into a phylogenetic tree. This is something that you should have done in Bio 20, but again, I know last year with COVID, I don't know how much detail you guys went into stuff. But basically, the more similar your DNA sequence, or more similar your gene sequence specifically, the more closely related the species are. So for example, the DNA of a house cat is a lot more similar to that of a lion than it is to a sparrow, because cats are mammals, sparrows are birds. Mammals and birds are two very different things, okay? This is also where you hear that humans are like 98% related to chimpanzees. This is because we've taken specific amounts of DNA and we've compared them, and those DNA sequences are 98% similar. But you also need to understand that we're related to corn by about 80%, okay? And houseflies by about, like, can't remember exactly, but another larger percent. So all of us have the same basic DNA sequences, so we all have the same bases, they're all doing the same thing, but we are all related because, again, we all according to evolution, came from a single-celled organism way, way, way back in the day, and that single-celled organism has turned into all of the life on our planet. Mutations do occur though, they happen in nature, they've happened over our history for many, many, many years, and then natural selection again is going to act on these mutations as to which ones are more beneficial than other ones. Again, which ones are going to give you a better chance of passing on your genetic information through having babies. There we go. So natural selection again is going to act on mutations. This is going to change the traits that are being passed down to future generations. And we also need to understand that mutations don't just happen in our nuclear DNA, so the DNA that's found in the nucleus, but chloroplasts and mitochondria also actually have their own DNA, and it's very specific to them. And mutations can also happen within these specific structures as well. And when we're looking at evolutionary history and evolutionary theory, mitochondrial and chloroplast DNA is actually super, super helpful with that because it's passed down in a slightly different way than nuclear DNA is. So mitochondrial DNA, for example, you only get mitochondria from your mom because during that whole process of fertilization where we have the sperm cell and the egg cell, when they come together, the sperm cell loses its tail and the mitochondria of the sperm is only found in its tail. That tail does not actually ever end up in the zygote. So the DNA that you have in your mitochondria is your maternal DNA. So it's mitochondrial DNA from your mom, and only your mom, and only ever your mom, for humans anyways. The fact that chloroplasts and mitochondria have their own DNA has led to a theory that these may have been their own fully functioning kind of cell-type things back in the day that were eventually incorporated into the cells that we have today, and they developed a symbiotic relationship with each other that were like, hey, we're both benefiting each other, so let's just continue to hang out. And again, that led to what we now know of as cells. So again, mitochondrial DNA is only ever coming from your mom. And scientists can take this mitochondrial DNA to help with studying human evolutionary history. Mitochondrial DNA is a lot shorter than nuclear DNA, so it's a lot easier for us to study than looking at the giant DNA molecule as a whole. So we do have two theories on human evolution. We have one called the multi-regional theory, and this one states that humans evolved at the same time in different regions of the world. So we had an earlier species, probably Homo erectus, and they dispersed to different parts of the world, and then they all happened to turn into Homo sapiens around the same time. This theory is not necessarily the more accepted theory when we're looking at evolution, but it is a theory that's out there, and there is some evidence behind it, so that's why it's called a theory. The theory that most scientists believe, though, is the monogenesis model. And so this one is that Homo sapiens moved out of Africa 2 times. So first, we had a mass movement of Homo erectus leaving Africa. So again, that's kind of like our precursor, according to evolution. And then the second time we moved out of Africa was as Homo sapiens. So we had developed into this new species, and we have now pollinated the planet as humans. Mitochondrial DNA studies support this monogenesis model, that every single Homo sapien on the planet did originate in Africa. And so when we look at mitochondrial DNA, the DNA that has the most variety or the most differences comes from people with African, or people who are currently living in Africa or have very close ties to African ethnic groups. Okay, so this theory supports the fact that mutations are going to build up over time, and the population that has been around the longest will actually have the most variation or the most differences or the most mutations in their DNA. And so people who live in Africa have the greatest amount of differences in their mitochondrial DNA versus people that live in Europe, versus people that live in South America, versus people that live in North America. So this again supports the fact that the African populations are the oldest populations on the planet, and that all humans at one point came from Africa. Okay, mitochondrial DNA found on other continents have also been traced back to Africa and not back to each other. So again, one more kind of nail in the coffin supporting the fact that all humans came from Africa. One other thing that scientists use when we're looking at evolution and when we're looking at similarities between species are things called interspersed elements. Again, this is like super complicated and I'm not going to get into a huge amount of detail. If you want to know more, take a course in university. Super fascinating stuff. But we have signs and we have lines. So signs are short interspersed elements and lines are long interspersed elements. And these elements are just giant pieces of DNA that don't do anything. And the evidence supports the fact that they're actually from viruses. So in the past, one of our common ancestors got infected with a virus, and that virus inserted its genome into that organism, and then it just continued to pass that genome along to future generations over and over and over again. So if we look at our DNA, we can say, hey, like 3 million years ago, our common ancestor had a really bad flu virus because we now have this viral DNA in our DNA. So we refer to these old viruses as retroviruses because they're old. And again, these lines and signs apparently come from these old infections. So a little funny for you, got your virus, and you got your retrovirus from the 70s, or like, 70 million years ago. So signs, again, these ones are short interspersed elements, so they're only about 300 base pairs, or 300 bases long, and they're very, very repetitive. So that would be like 300 bases of A's, or 300 bases of G's, or 300 bases of C's just hanging out. And when we're like analyzing your DNA and you get to this giant segment and all of a sudden it's C, 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 scientists are like, huh, that's kind of weird. And then they look into it. We also have lines, which again are the longer interspersed elements. And this is where we have repeated DNA sequences that are 5,000 to 7,000 bases long. So again, when we're analyzing this DNA and all of a sudden we get to an area and it's like the exact same sequence over and 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 over again, scientists get a little curious about the reasoning behind this, and then they start to compare them between different organisms. And we can then see, based on these signs and based on these lines, again, how closely related certain animals could be. Because if they had a common ancestor that ended up with this virus, and then that common ancestor split into the two different species, so let's use human and apes for example. Okay, humans and apes should have these specific DNA sequences at certain points in their DNA because their common ancestor also had this virus. So again, these just help to give scientists a little bit more support for their evolutionary theory. And again, it's a lot more complicated than I just said. I'm just giving you like the short and quick version of it. So if two species have the same sign or line at the exact same position of their DNA, again, we assume that this insertion only occurred once in a common ancestor that was then passed down to the future species that were created. So signs and lines, again, are ideal as evolutionary markers in DNA. And they're also ideal because they're very long pieces of DNA, and again, they're super recognizable because again, when a scientist is looking at the DNA sequence and all of a sudden we get to this huge line of the exact same DNA, you're just like, huh, that's weird. right. So mutations again, lots of different types. Harmful, they're bad for you. Beneficial, they're good for you. Neutral, meh, they're fine. These mutations again can be broken down into point mutations. So silent, again, those ones would be neutral. They don't really do anything. Nonsense, turning it into a stop mutation. Again, we say that this is typically lethal. So this one could technically also be considered a harmful mutation. And missense is taking an amino acid and turning it into a different amino acid, which could be either beneficial or harmful depending on how it impacts that organism's ability to reproduce. Gene mutations, again, insertion and deletion, putting nucleotides in, taking nucleotides out. Translocation, taking a piece of DNA from this point, moving it over here. And inversion is taking out this piece of DNA and just flipping it backwards and putting it back in the same spot. All of these are again going to lead to frameshift mutations, which is where we have a difference in the amino acid sequence and we're changing the reading frame of that protein. And all of these mutations can lead eventually to the creation of different species over time if there's enough of them. And the mutations that we have and different parts of our DNA that can be found can also help us kind of figure out the evolutionary history of the organisms on our planet. Again, signs and lines come from old viruses that show us kind of common ancestry between species. And then we also have mitochondrial and chloroplast DNA. Mitochondrial DNA, again, comes only from your mama, not your dad. You got it from your mom. So that is it for Unit C. Next week, we will be finishing this, and then when you guys get back after break, we get to finish Unit D, talking a little bit more about genetics, but how that relates to populations.